giving me this opportunity to talk on biomarkers of Parkinson's disease. And I also wish to welcome anybody and everybody who's here, delegates as well as patients, to Center for Brain Research. So what I will do in the next 15 to 20 minutes would be to kind of run through and give you an overview of the various biomarkers of Parkinson's disease. Now, this is a fairly extensive topic, so I'll restrict myself to just fluid biomarkers as well as imaging biomarkers, which is what I usually work on. So quickly starting with the presentation, there are quite a lot of fluid biomarkers. Researchers have exploited almost every biofluid that is available in the body to identify biomarkers for PD. But the most common ones that you could find would be cerebrospinal fluid and plasma or serum. The cerebrospinal fluid, while it adds a lot of value because it is so closely invested with the central nervous system, the plasma and serum markers are better because they are non-invasive and doesn't require lumbar puncture to perform the studies. While there are a few studies on saliva, tears, as well as urine, none of the studies have actually provided a biomarker that have withstood the test of time. So you have more of CSF biomarkers or plasma biomarkers, and that is what we are going to discuss. Now, there are two ways in which biomarkers are usually measured. So either you measure for a very specific molecule, which is considered a biomarker, or in the recent times, we have also started using high throughput techniques and high throughput screening to identify a large number of biomarkers in a single sample. Now, what I will do is talk about just few of these biomarkers that is quite well validated. Now, I cannot call them as biomarkers per se because they do not hold every time and in all conditions, but these are all potential biomarkers to be exploited in further studies as well as for patient care. Now, the most common one is alpha-synuclein. Alpha-synuclein, as you know, is a presynaptic protein and is considered to be a molecule which is aberrant in Parkinson's disease, and they tend to aggregate and then deposit to form Lewy bodies, which are the pathological hallmarks. You can see a Lewy body here. So these are the pathological hallmarks or histopathological hallmarks for Parkinson's disease. So these are essentially intracellular structures, intraneuronal aggregations of alpha-synuclein. Now, it was logical for scientists to think that if alpha-synuclein is depositing or it is in the core of the pathology, why not measure the alpha-synuclein levels? So this has been done. So scientists have measured CSF levels of alpha-synuclein as well as plasma or serum levels of alpha-synuclein. And it kind of shows a pattern in which in most of the Parkinsonian disorders, including Parkinson's disease, dementia with Lewy bodies, multiple system atrophy, cortical basal syndrome, or progressive supranuclear palsy, the alpha-synuclein levels tend to decrease in the CSF, which has been detected and been used as a biomarker in many conditions. At the same time, in plasma and serum, the levels of these proteins tend to increase. This is primarily because the entry point of these proteins into blood is usually through the blood-brain barrier or due to increased expression of these proteins in the periphery. Now, while CSF as well as plasma serum alpha-syn was considered to be a decent biomarker, we also realized that the oligomeric and the phosphorylated alpha-syn isotypes or isoforms are probably more important because these are the parts which kind of aggregates together and then clumps and deposits. So studies shows that if you measure, instead of using the total alpha-syn, if you measure oligomeric and the phosphorylated alpha-syn, the sensitivity of diagnosis of synucleinopathies, which includes PD and MSA, goes up to 95% and 98%, which is very high for diagnosis. And it also offers a discriminatory potential of 88 and 96% when you try to discriminate PD from other atypical Parkinsonian syndromes. Now, with advent of newer high throughput assays like PMCA or RT-QuIC, we hope that we will be able to pick up oligomeric and phosphorylated alpha-syns in extracellular vesicles and as well as deposits in skin or even in other biofluids much more efficiently. So this is a very interesting time to be a researcher for biomarker in alpha-synuclein. Now, alpha amyloid beta and tau proteins are typically markers that you would consider in Alzheimer's disease and other cognitive neurodegenerations. But there are many studies which shows that the A beta proteins and tau proteins tend to interact with alpha-synuclein, and both will increase the chances of aggregation. Consequently, people started looking at amyloid beta and tau proteins as well. And again, it showed that in the CSF levels, the amyloid as well as tau both were decreased, whereas in plasma, it was slightly more increased. But as of now, studies have not been able to identify whether these are very specific for Parkinson's disease or are they more associated with the cognitive impairment that happens with PD. So PD has two states known as PD MCI and PD dementia, which happens in a fraction of patients. Studies shows that if there is a dysregulation of amyloid beta and tau, it is most likely an indicator that the person with Parkinson's disease is developing cognitive impairment. And hence, amyloid beta and tau proteins are considered to be potential biomarkers to highlight or understand the cognitive trajectories in Parkinson's disease. The third and probably very much upcoming biomarker is neurofilament light chain. Neurofilament light chain is basically the cytoskeletal structure of neurons. So anything that happens to neurons leads to leaching of these cytoskeletal structures into the CSF and then into the blood. So anytime it increases, that usually means there is some kind of neuronal injury. Now, if you look at the graph, the histograms that is given, everything is written in short form, but you have Parkinson's disease patients, you have Parkinson's disease, PD dementia, Alzheimer's disease, dementia with Lewy body, normal pressure hydrocephalus, frontotemporal dementia, traumatic brain injury, PSP. Virtually almost every neurological disorder associated with neurodegeneration shows twofold to almost 40-fold increase in neurofilament light chains. Consequently, NFL is not considered to be a specific biomarker for Parkinson's disease, but associated with non-specific neurodegeneration. When you combine this NFL with other Parkinsonism-specific neurodegeneration, there is a potential for actually changing the time of diagnosis from a clinical to a prodromal or preclinical stage. That is why NFL has become an important one. Now, there are many other biomarkers that has been identified through multiomics and high-throughput techniques. I'm not going into any of these things. So in the table, I put those proteins which have been identified as potential biomarkers in at least 10 studies. So you have all of these, and if you look at the conditions under which they were studies, you can find that you have both PD, you have AD, you have non-specific neurodegeneration, all of it. So probably in the next 5 years or 10 years, you could see many of these biomarkers coming into clinical scene as well. In addition to that, there are some other techniques. I told you that there are you can either identify specific molecules or you could use some high throughput techniques. So this is an example of a paper from Dr. Falguni Alladi's lab, in which we worked. What we did was we used CSF from patients with Parkinson's disease and normal pressure hydrocephalus, performed mass spec-based proteomics, and then identified all the upregulated and downregulated enzymes. And we found, it's not very clear there, but we found a total of 28 proteins which were upregulated, out of which fibrinogen and complex factor H were considered to be very good at discriminating PD versus other non-Parkinsonian phenotypes. So there is quite a lot of potential for all proteomics, lipidomics, metabolomics, glycomics, transcriptomics, as well as genomics as biomarkers for Parkinson's disease. Now, quickly jumping from fluids to radiological biomarkers. So almost every radiological modality has been used as some kind of biomarker or the other. So you have CT scans, which show some kind of biomarkers. You have ultrasound, which has been performed in Parkinson's disease, which also is considered to be a biomarker. But I'd like to restrict to the common ones, which are the magnetic resonance imaging, as well as the PET and SPECT, which are molecular imaging. Now, this is probably the most common way a clinician would kind of diagnose a case of Parkinson's and differentiate it from other atypical Parkinson's. So this is all figures of what an MRI looks like in different kinds of atypical Parkinson's. And so if you see this part of the brain which I am highlighting, that is the hindbrain structures that kind of looks like a hummingbird or a penguin. So that is known as a hummingbird or penguin sign in PSP. You see this, you know that there is something wrong with the midbrain, which is a sign of progressive supranuclear palsy. The same thing, when you look axially, you can see a Mickey Mouse-like structure here. That's another pathognomic mark. In another synucleopathy known as multiple system atrophy, you can see this particular cross-like structure on the pons, which looks like hot cross bun. right? And based on this, clinicians can kind of figure out what kind of neurodegeneration you're dealing with. But again, these are not considered true biomarkers because when you see the sign, the person has already developed a lot of clinical symptoms. So only in diagnostic confusions you would use this as a biomarker. Rest of the time, it is, based on the clinical symptoms, it is quite apparent that it is Parkinson's disease. Now, there are some planimetric techniques where you could measure different areas of the brain, different parts of the brain, create ratios, and then identify, differentiate different types of atypical Parkinson's. This is just an example of something known as midbrain indices, which is used in progressive supranuclear palsy. But again, these are not true biomarkers, but these are very good clinical markers to use in clinical practice. Now, coming to the biomarker part. All right. It's not moving. All right. So in the last 10 to 15 years, we have had some biomarkers in neuroimaging specific for Parkinson's disease. One is known as detection of nigrosome 1. So if you look at this image, you can see a white structure here. This structure is known as nigrosome. It kind of represents a body which has 40 to 50% of total dopaminergic neurons in the brain. So once that white part kind of starts disappearing, as in this case, you would consider this as a sign of Parkinson's disease. Again, the sensitivity of this detection is only 95% and pooled sensitivity specificity of 90%. So when you compare nigrosome 1 in different atypical Parkinsonism, the sensitivity and specificity comes down significantly. You have another form of MRI known as neuromelanin sensitive imaging, in which you kind of quantitate the amount of neuromelanin that is present. So this is Parkinson's disease, and this is healthy control. You can see a thick band of white here in control, whereas on this side you can find that it has come down. So that also signifies Parkinson's disease. And if you, this is not very clear, but if you see the white dot here, you can see that at the arrow, the white dot has come down significantly. So that is also a loss of neuromelanin in the brain, and it is also considered to be a biomarker for individual cases with Parkinson's disease. Now, visually looking at this is one thing, and when you are looking for a study or a biomarker, you would obviously do contrast ratio scans and additional sequences and volumetry to give a value based on which you would research. Now, just like neuromelanin quantification, iron quantification has also become a very important biomarker in Parkinson's disease. The basic idea is that, so the basic idea is that when there is degeneration, iron deposits increases. So in iron quantification, what you're essentially looking at is how much of signals are picked up by the iron molecules. So in this image, you see Parkinson's disease, you see that the substantia nigra is quite dense, whereas in this one, it is low. So that is also now considered to be a biomarker for Parkinson's disease with reasonable sensitivity as well as specificity. But then if you look at what is written below, most of these can also be found in other synucleinopathies, in AD, as well as other atypical Parkinsonian syndromes. Consequently, much more of studies are required in these fields for diagnosing PD. The last two scans would be on SPECT. SPECT is a functional scan. It basically involves giving a radio tracer that will detect dopamine transporter. So what you can see here is a patient of Parkinson's disease. So there is a significant uptake here. But that when compared to the various patients of dementia with Lewy body or essential tremor or Alzheimer's disease, you can see that there is a significant variation. And this variation is also considered to be a diagnostic biomarker. DAT scans are very commonly used in clinical practice as a biomarker even now. The last one would be PET scans. PET scans are positron emission tomography. Essentially what you do is very similar to SPECT, except for that you have a radioligand that is pushed in. The radioligand goes into the brain, and then metabolism and its uptake are usually detected. There are usually two main types. You have the PET scan, the FDG PET scan, which basically use glucose. It is not considered to be a great biomarker because in PD it is almost always normal, whereas in all the other disorders it is quite abnormal. So if you want a differentiation between PD and atypicals, this is what you use. And the last one is PET. You can see the uptake that has happened, and the loss of the red signals in Parkinson's disease. I think with that, I'll just summarize. There are different types of biomarkers. You would use all biomarkers for different purposes, ideally stage depending on which stage of Parkinson's disease you are dealing with. My acknowledgements: I am funded by the Scan Research Trust for my work in Parkinson's disease, the CBR funds for work in age and age-related neurodegeneration, and Pratiksha Trust for work on atypical Parkinson's. Thank you.
Thank you, Dr. Albert. We can have 1 or 2 questions from audience.
How easy or difficult, because by CSFs is the most difficult samples
Yes.
to be collected, right? Even
Yes.
the blood samples, it looks like the sensitivity and specificities are low. Probably the way we pick up should change,
Yes.
because if we can try to pick up more from the blood samples would be the best way to go forward.
Yes.
So what do you think, where are we standing at that point of time?
So at this point, it depends entirely. The reason why CSF is a beneficial thing is because obviously the pathology is in the brain. And the second thing is the amount of change would be more in CSF when compared to blood. So as long as we have techniques which can measure the same molecules in blood with much higher sensitivity, it is possible to do. For example, at CBR, we have a single molecular array which can detect at femtoliters, and that is much more than what a routine ELISA can. So we are definitely at a point where we can detect things from CSF in blood, but the problem is we always need to first prove that the changes in brain is reflected in CSF and the changes in CSF is reflected in brain. So this is a very hot area of research as of now. But yes, blood would be the most ideal marker.
I think that is where most of the things are standing, right?
Yes.
So somewhere we need to start more into other blood biosamples, and quickly should be able to prove that
Yes.
works out.